Hi, in this video we'll talk about 3 different ways that the cell repairs damaged DNA. In method 1, we'll talk about base excision repair. This is when a single base is affected. In this case, there's a type of damage called deamination. This is when a C base within an amino group loses it's deaminated, and then you have a base which has a carbonyl group in place of the nitrogen, and coincidentally this base is uracil, the base that is in RNA. Now you'll remember that uracil acts the same, base pairs the same as a T, and so this seems to the DNA like a C to T mutation. But there are also special enzymes, for instance uracil DNA glycosylase, that recognizes that uracil is not to be incorporated into DNA. It's an RNA nucleotide. And so it'll scan the genome for places where a U is present. When it finds it, it will excise the base, then cut the backbones of the sugar and phosphate groups, insert the correct base, which is done by DNA polymerase, and then DNA ligase will seal the nick. There is another type of DNA damage called depurination, which happens to purines, which are G's or C's, and those affect the bond between the sugar and the base, and the bases will be lost, and so those types of mutations or DNA errors enters this flowchart at this position. So these are both 2 very common kinds of mutations, and they are repaired effectively by the cell. However, not perfectly. We can actually see evolutionarily that there are more C to T mutations than other types of base pair changes. So again, it's a very common occurring event, and the cell catches most of them. A second type of method is called nucleotide excision repair. In this case, 2 or more bases can be affected, and the type of damage shown here is a pyrimidine dimer, which is a more generic term for the type of damage we saw in the previous video, thymine dimers or thymidine dimers. So this is a pyrimidine dimer. Instead of TT, we have a CT base pair. And once again, there are proteins which constantly scan and surveil the genome. When it finds this kind of an error, it will cleave the backbone at some distance from the lesion site, remove the entire piece of DNA using the enzyme DNA helicase. DNA polymerase will fill in the template 3' to 5', and ligase will seal the final nick. So again, this is an efficient mechanism for repairing thymidine dimers. So now we're going to ask, before we get to the third mechanism, we're going to ask the question, how does the cell know when it encounters a bubble or a mutation? How does it know which strand to repair? This picture is not very realistic because really the bubble is on both sides. It sort of implies that the correct base is a straight and normal backbone, and then because you have an incorrect base it bubbles out a little bit. But obviously that's not the case. After replication, a T will be inserted next to the A, and a C will properly be inserted next to the G, and going on you would have 50% mutated cells and 50% non-mutated cells. Now although we don't know how yet, if the cell knows somehow that the A is the incorrect base, it can correct that, and then it can put 2 G's there, and now you'll have 100% correct of the cell's progeny will have the correct genome. But think about the consequences if the incorrect base is repaired. If the G is cut out and it gets converted to a T, now you have 100% of the progeny cells having the mutation incorporated. So how does the cell figure out which is the proper base to go at that position? The answer is quite ingenious. And what the cell does, which gets it right most of the time, but not every time, is it will, once it finds a mismatch here, it will scan in both directions, looking for the closest nick in the backbone of the strand. The cell then assumes that this is the most recently synthesized strand, the other strand with no nicks, having stood the test of time per se. And so it will decide to cut out the region of the DNA that has the pre-existing nick, and it will repair that again with, I should draw it like that, with a polymerase and then ligase sealing the final nick. As you might guess, this all happens relatively quickly if right after DNA synthesis, because if a different enzyme happens to notice there's a nick present in the DNA, it will repair that, not knowing that there's a bulge there. So after synthesis, the DNA there's a basically a ticking time bomb where the cell has to repair this bulge before another enzyme fixes this nick, if it's going to use this mechanism for picking out which strand to repair. Now, the third mechanism that we will study is a mechanism called homologous recombination. And this is the way that the cell repairs, most of the time, the most serious of DNA lesions, that is a double-strand break. I suppose it should talk about non-homologous end joining first, and that is when a double-strand break is identified. If it cannot use homologous recombination, it will just cut back the ends of the DNA to blunt ends, jam them together. You have deleted DNA sequence for a protein that could be disastrous, whether even a single base, because it will change the reading frame. And that's better though than having a broken chromosome, but this is a sort of a last resort. The method we'll talk more about is homologous recombination. And what you have to remember is that you have two copies of every chromosome. You have one chromosome from a father and one chromosome from a mother. And what it's going to do, it's going to, again, cut away the affected region. And what it'll do, it'll synthesize new material. It's going to be direct copies of your maternal copy of the chromosome. So it will change your genetic material, but hopefully it won't incorporate a deleterious mutation. And this is a far preferable alternative to simply jamming the two ends back together. So this is the mechanism for homologous recombination, and in this example we have a double-strand break, and the first thing the cell does is it will use an exonuclease to chew back in the 5' direction. The purpose is to create a segment of the DNA which is single-stranded and then available for base. It miraculously aligns with the other parental chromosome and now is part of a double helix here, and it can go on synthesizing in the normal 5' to 3' direction, where the bulge of the other strand of the paternal chromosome is being successively displaced until it has traveled some distance. Now it has enough material to reach the existing maternal chromosome, and then ligase can seal that nick. At the same time, the existing part of this strand now can be used as a template for synthesis of the other strand, and again, ligase will seal the nick here. So overall, I think this is pretty amazing. One of the ways double-strand breaks recur is through ionizing radiation, like X-rays or cosmic rays. But a lot of us don't get exposed to X-rays anyway that often, but a common way that double-strand breaks form is when a replication fork encounters a nick in the backbone. If you have leading strand synthesis and you encounter a nick, that's it, that's done. And there's no further extending because you no longer have a template strand that is in the proximity. The cell will begin homologous recombination by using the exonuclease activity in the 5' direction and having a strand that can base pair to the other chromosome and start the repair process. We will encounter homologous recombination later in the course when we're studying meiosis. And you may already know, but we will learn that although you inherit one chromosome from your father and one chromosome from the mother, the chromosomes that you pass on to your offspring are not purely your paternal or your maternal one, but a combination that is unique to you. And in fact, it's very nearly 50-50 in each of your offspring. So I'll just spend a few seconds showing you a mechanism. How is it that in some cases homologous recombination can affect the exchange of genetic material versus simply resolving it as a repaired segment? The key to this is to focus on this particular intermediate called a holiday junction. That's a rather famous term. And the difference is how these intermediates get resolved. If we cleave this chiasma this way, you will have an exchange of genetic material. If you cleave this intermediate that way, you will have a repaired chromosome. It's impossible to visualize here. Let me try to make it a little clearer. This is the form that was shown on the previous slide, and what I'm going to do is I'm going to unwind this in this direction, and now we have an open form, and I think you can see here, if you cleave it that way, that in one case you'll have repaired material, or in the other case you'll have translocation with some of the paternal and maternal material. And that is made even clearer here on this slide. So that concludes the 3 different types of DNA repair that we're going to talk about: base excision repair, nucleotide excision repair, and homologous recombination. Please answer the questions at the bottom of the page. Thanks.